everybody. Hope you all are fine. Today we are going to start the development of the foregut, its malformations, and the genetic regulation of the gut tube. This topic has been divided into three parts. So at the end of part 1, the participant should be able to describe the origin and development of the gut tube, classify the gut tube derivatives, describe the morphogenesis of the gut tube, and discuss the genetic or molecular regulation of the gut tube. In part 2, we will describe the morphogenesis of the esophagus as well as the stomach, malformations of the stomach, and rotation of the stomach, mesenteries of the stomach, and omental bursa. Whereas in part 3, we will discuss the malformations of the stomach, morphogenesis of the duodenum, and malformations of the duodenum. We start with the gut tube formation, which occurs because of folding of the embryo. This folding is divided into cephalocaudal folding, which occurs because of rapid growth of neural tube, especially the brain, as well as rapid lengthening of the embryo. Number 2 is lateral folding, which occurs because of rapidly growing somites and spinal cord. This leads to formation of right and left lateral folds, the primordia of the ventrolateral body wall. The end result of these folding is the final appearance of the embryo as a cylindrical structure. But remember one thing, these folds occur simultaneously and are not separate sequential events. As regards the outcome of the folding, if we compare the upper three diagram A, B, C, we can appreciate that reorientation of different structures occurs, but the sequence of these structures remains same. And the best example seen is cardiac area, which is initially in the cephalic region or in the cephalic part, which comes to lie on the ventral aspect of the developing embryo. Number 2 is expansion of the amniotic cavity at the cost of extra embryonic coelom. Number 3 is reduction of area of communication between intra and extra embryonic coelomic cavities. And number 4 is embryo becomes enclosed by the ectoderm. But remember, the main result of this folding is incorporation of an endothelial line part of the yolk sac inside the body as the gut tube. Now we come on to the derivatives of the gut tube. Towards end of 4th week, a tracheal diverticulum, which is also named as lung bud, appears on the ventral aspect of the cephalic part of the gut tube and divides this tube into a cranial segment, which is also named as pharyngeal gut, and ultimately develops into various parts of the head and neck, and a caudal segment, which is the gut tube proper. This gut tube proper, depending upon the its blood supply, is divided into three parts: foregut by celiac artery, midgut by superior mesenteric artery, and hindgut by inferior mesenteric artery. The endodermal lining of the gut tubes ultimately gives rise to number 1, epithelium of gastrointestinal tract, respiratory tract, urinary bladder, and most of the urethra, tympanic cavity, tympanic antrum, and auditory tube. Number two, it also gives rise to parenchyma of thyroid, parathyroid, tonsil, and thymus glands, and liver and pancreas. Now let us tabulate the gut tube divisions and derivatives. As earlier said, the gut tube is divided into two parts, cephalic and caudal, by the appearance of lung bud. Cephalic part develops as head and neck region, whereas the caudal part is named as gut tube proper and is divided into three parts depending upon the blood supply of each part. First part of the gut proper is the cephalic part, which is also named as foregut. It is divided into three parts: the proximal part, which develops as esophagus, middle part, which develops as stomach, and distal part as first 1.5 part of the duodenum. The second part of the gut tube proper is the middle part, which is also named as abdominal midgut. It has got two limbs. The cephalic limb develops as next 2.5 part of the duodenum, whole of the jejunum, and major part of the ileum, whereas the distal limb of the midgut part develop as terminal ileum, appendix, cecum, ascending colon, and right 2/3 of the transverse colon. The third part of the gut tube proper, the caudal part or the hindgut, is again divided into three parts. Proximal parts develop as left 1/3 of the transverse colon, middle part as descending colon, and the last, the distal part of the caudal part, develop as the sigmoid colon, rectum, and upper 2/3 of the anal canal. Now we come on to the morphogenesis of the gut tube. First, the morphogenesis. It is the process which involves origin and development of different morphological characteristics of an organ as form, shape, and structure. Morphogenesis involves different processes such as number one, elongation and looping of an organ because of differential growth in the walls of that organ. Number two, herniation of the parts of the gut into umbilical stalk and withdrawal into the abdominal cavity because of organ growth disparity and available space in the abdominal cavity. Number three, rotation of several local regions of the gut because of differential growth of the walls and growth of surrounding structures. Number four and final, histogenesis and functional maturation. The process of morphogenesis is regulated by the genetic control of the body. Now we come on to the genetic or molecular regulation of the gut tube. The regional specification of the gut tube depends upon reciprocal interaction between the endoderm and the mesoderm present in the wall of the gut tube, and it occurs when the lateral body folds are being formed and these folds are approaching each other to form the gut tube. This prefiguring involves number one, craniocaudal patterning, number two, radial patterning, and number three, luminal formation. Prefiguring in craniocaudal patterning involves formation of two specific zones. Number one, specific regions such as esophagus, stomach, and intestine, etc. Number two, important junctions such as gastroesophageal, gastroduodenal, and ileocecal junctions, etc. This patterning, that is craniocaudal patterning, is initiated by retinoic acid, which is present in the wall of the gut from pharynx to the colon, and is established by nested expression of segmental, selective or combinatorial codes of FOX transcription factor in the mesoderm. And this transcription factor is induced by the SHH gene present in the endoderm. These codes cause a genetic cascade that regulate the specification of each gut region, such as SOX2 specifies esophagus and stomach, PDX1 duodenum, CDXC small intestine, and CDXA specifies large intestine and the rectum. In radial wall patterning, the gut tube wall differentiation occurs under the influence of reciprocal interaction between small acid proteins and bone morphogenetic proteins. These influence as the high expression of SHH leads to certain Hox codes, which number one inhibit smooth muscle activating protein, which in turn inhibit the smooth muscle differentiation. These codes also induce BMP, which inhibit neurogenesis. Low expression of SHH induces small acid proteins which promote smooth muscle differentiation and inhibit bone morphogenetic proteins which promote neurogenesis. Concentric layers of Hox codes are accomplished largely via the expression of SHH in the endoderm. Initially, more SHH is present close to the endoderm, which inhibits smooth muscle and neuronal differentiation. Further away from the endoderm, SHH concentration is lower, which permits smooth muscle and neuronal differentiation as muscularis externa. Later on, when SHH expression goes away, it allows development of smooth muscle as muscularis mucosae and neurons of the submucosal plexus. Number three, lumen formation of the gut tube. As the gut tube develops, endoderm proliferates rapidly and occludes the lumen during 5th week of development, and this occlusion is named as epithelial plug. Growth and expansion of the mesoderm components coupled with apoptosis of the endoderm during 7th week of development leads to production of vacuoles and ultimately recanalization during 9th week of development. In this way, the lumen is restored. Apoptosis and recanalization process is governed by smoothant proteins which are encoded by SMO gene. This occlusion and recanalization process occurs throughout the gut tube, and any error in this process anywhere along the length of the gut tube can result in stenosis, that is narrowing or occlusion in that very region. To be continued as part 2.